So I guess this is a shortish introduction to phylodynamics. I thought this is quite a non-technical presentation. There were no equations or anything. I thought I'd just go over kind of what phylodynamics adds to phylogenetics, and also give some examples of previous papers, mostly in HIV, and what they bring to the field. So I thought I'd start just with a quick recap on phylogenetics to make sure everyone's up to speed on that. So we're talking about the study of evolutionary relationships between organisms. And in the cases we're looking at, the organisms, the pathogens. The kind of guiding principle behind all of phylogenetics is that the greater the similarity is between two organisms, the more closely related they are. And hence, that means they more recently shared a common ancestor with each other. And that data, that principle using genetic data is used to construct phylogenetic trees that depict the relationships between the ancestors of your samples. Usually these days, especially in pathogen studies, DNA or RNA sequence data is used as a genetic data to look at distances and construct relationships. And next generation sequence technology is making the acquisition of molecular data much faster and cheaper than it used to be. So this is an example phylogeny which I just randomly generated in R, which the features of which are it has nodes, so which are all where two branches join is a node, and there are two types of nodes. There are external nodes or tips, which represent the samples in your data set and appear right at the end of branches. And there are internal nodes, which represent the unsampled common ancestors of organisms in your sample. Some, but not all trees, will have a particular internal node called a root node, which represents the common ancestor of every sample you have. And time sort of implicitly goes from left to right in this diagram, which I normally go as either left to right or top to bottom. So the node on the right is the node on the left is the root one, ancestral to your whole sample. Branches connect the nodes, and the length of those branches is some measure of difference between the two nodes. It's either some measure of the number of mutations that have taken place, or it's some measure of time, which I'll talk about shortly. So longer branches mean there's more mutations happened or more time has elapsed. And a clade is basically a node and all its descendants. So the red highlighted one there is one node and all the internal and external nodes and their branches connecting them. So branch lengths in many phylogenies, perhaps most phylogenies in literature, do represent genetic distance, which is a measure of the amount of mutation that has happened over that branch. Often that's for DNA and RNA, that would be in substitutions per site. And there are lots of methods and packages that you can use to create phylogenetic trees with branch lengths in genetic distance. So neighbor joining and maximum parsimony, you might have heard. Maximum likelihood is probably the most common you see these days. So when trees are made in FIMO, VAXML, or IQ tree, those are maximum likelihood. There's also fast tree, which is a very, very fast program, but it's an approximate maximum likelihood method. There's the other category of main category of packages are the Bayesian packages, which 3 are listed there. And one thing to always bear in mind when doing phylogenetics is that that uncertainty exists in the phylogeny. You can never know that your phylogeny is the correct ancestry unless you observe that ancestry, which we normally don't. In fact, there'd be no reason to, but then you wouldn't need to build the tree. And sometimes the data makes many separate ancestries about equally likely. And so there needs to be some way, ideally, an analysis to quantify the uncertainty surrounding the reconstruction. It's handled normally by one of two processes which in some ways add up to something quite similar. So in bootstrapping, which is used in maximum likelihood and other and similar methods, the tree is constructed several times using subsets of genetic data, and then you get a set of several bootstrap trees. Whereas Bayesian methods kind of, as a natural output, they give you many rather than one tree. they aren't just finding the maximum likelihood tree, they're trying to find a representative sample of trees compatible with the data. So we can get out many trees from that. And so in either case, you get many trees, and often papers will present a summary tree, like the one on the right, which is a tree of HIV and SIV sequences in a paper on the origins of HIV. You'll get one tree with numbers, which are bootstrap numbers here, of how many of the 100 bootstraps they did agree that clade existed. So the higher the number, the more confidence there is in the ancestry going down from that node. Phylogenetic analysis of a single tree, therefore, is not ideal, but sometimes it's the only choice. Sometimes there's no way you can accommodate uncertainty. So the phylogeny reconstructs the ancestry of the samples you have, but on its own, it can't date the events indicated. So if you've got a tree like this, you think it's the right tree, you know what order the ancestors existed in, but you don't know when those ancestors existed. You have no idea of how the length of the branches, if they're in mutations, translates to the length of the branches in time. But what we'd like to be able to do is to be able to date those internal nodes, so we can then estimate when common ancestors existed, and that implies when transmissions would have occurred between individuals. If we have is samples that were collected over quite a short period, then there's not much we can tell from that, and we're going to need some other information for dating. So for macroorganisms, and particularly for humans or any other large animal, normally breeding and mutation is too slow for measurable changes to be observable over a useful timescale. If you were to take a sample of humans this year and one 5 years ago, there wouldn't really be a noticeable difference in the genetic makeup of the population. So studies of that kind sometimes have to use ancient DNA or other archaeological or paleontological findings to time the tree instead. But the nice thing about pathogens is that their lifespans are very short, and many of them, especially RNA viruses, mutate very fast. So populations observably evolve over short timescales. And what we can do is we can use the difference in the sampling dates we've got to estimate the rate of mutations without having to appeal to old DNA or to some other reference point. So these are the same tree, the same data set, which is a set of dengue virus sequences. It's part of the BEAST example data sets. On the left is an undated tree. So the tip names have 2 digits at the end, which are the year of sampling. And you can see that as you go from left to right, they do generally increase, but it's not that they perfectly increase. You can see that the 1982 one is further to the right than the 1983 one, and then the jump here from the 86 to the 94 is not it doesn't look like 8 years compared to the other distances. So that's a tree where the mutations are in substitutions per site, and you have a scale bar here. Whereas on the right, you have the same sequences, but fit to a timeline. So you have a calendar timeline along the bottom, and here all the tip dates will actually correspond to a point on that, so they're exactly matched. It's taken those dates and fit the tree so that it fits there, and looked at how long the branches should be under calendar time. But mutation is not a deterministic process, so a longer phylogeny branch does not automatically put a sample further forward in time. You can see that again with the 82, 83. 82 is further back in time, further forward in mutations. So mutations are instead usually assumed to occur at a particular rate per unit of time according to what's called a molecular clock. It's determined as the rate at which mutations occur. The simplest version of the molecular clock is called a strict clock, and that assumes that the rate at which mutations occur is constant over time for a given sample. This is usually thought to be an oversimplification, so there are various forms of relaxed clocks that allow rates to vary in different regions of the phylogeny under various different models, which I won't go into now, but there are quite a lot of different versions of a relaxed clock that have been allowed. It's important to notice that not all data sets will display behaviour consistent with a molecular clock at all. You may find that there's no particular correlation between how, if you've rooted the tree, how far your tree is the root, and how when it was sampled. And that may be because recombination or convergent evolution, for example, have kind of mixed up the signal and you can't get it. Or it may just be because you don't have enough variation in your data set to establish the signal. So it's worth checking for this if you're doing analysis with a molecular clock. And the presence of a signal, there's a program called Tempest, which is a very, very, very quick way to see if there's any temporal signal in the data set. So then I bring in phylodynamics. The term was coined by an article by Brian Grenfell in Science in 2004, was the melding of immunodynamics, epidemiology, and evolutionary biology. As it's turned out, the immunodynamics bit has been a bit of a separate stream, which maybe could be talked about another time, and I'm going to talk about the kind of the evolutionary aspect. There's not been much work that's really combined the immunology and epidemiological aspects of it. But the principle behind the epidemiological version is that a data phylogeny can be seen as a partial history of the set of pattern lineages you've got in your sample. So the tips represent samples, and the internal nodes represent the common ancestors of the tips, which are the common ancestors of pathogens infecting the hosts that you've sampled. And they often assumed to also represent transmissions, roughly represent transmission between two hosts. That's not to say that they're transmissions between the two hosts that you sampled, just as when you're doing phylogenetics, you're looking at unsampled common ancestors that aren't necessarily organisms that you know about. Similarly, these are events that split the lineages, which in an epidemic would have to be a transmission, but it wouldn't necessarily have to be a transmission between sampled hosts. The sample dates you have from your data, then you use to calibrate the timings of those ancestors or equivalent transmissions according to a molecular clock. And then you've got this timed phylogeny, which is a history of an epidemic or of an endemic disease. And if you then you find a mathematical model of the process that generates that tree, you can use sequence data to estimate the parameters of that model and then learn about pathogen dynamics. So there are, I'd say, probably broadly three classes of mathematical models that are commonly fit to time trees. There are population genetic coalescent models. There are forward time epidemiological models, which are more traditional kind of compartmental mathematical models of transmission. And there's the epidemiological coalescent models as well. A couple of other topics I'm going to touch on are phylogeography and the reconstruction of the epidemic transmission tree. So population genetic coalescent models, the principle here is that if you were to pick two individuals of one generation of organisms at once, if they're in a small population, they have a much greater chance of sharing a common ancestor in the previous generation, but there were just fewer people in that generation to have given birth to them. So the idea being, if you look back at the tree and two branches take a long time to coalesce, they were probably in a large population, but if they take a short time to coalesce, they were probably in a small population. This is assuming that free mixing of organisms, which is usually violated in practice, but it can be a recent generalization. And then what you can do is you can use the distribution of internal node times, how quickly lineages meet a common ancestor, to learn about the size of the population. And the right is an example from a review by Denise Kunert and Desi Wu about 8 years ago, showing that on the left-hand side you have a static population of a similar size, and on the right-hand side you have a population that's growing exponentially. And you can see that as the population shrinks, the coalescences get closer and closer together, because so your date of your root of the tree is closer to the present. So the simple case, as in the left-hand diagram here, is the population size is assumed to be constant. You just estimate that size, although because of the assumptions in this model, it's quite difficult to translate what's called the effective size, the effective population size, to a real number. Alternatively, you can assume that the population is obeying some kind of parametric function, such as exponential growth, like on the right there, or logistic growth, or something else, or kind of step functions. But probably best of all is you can divide the timeline and estimate separate size in each period. So on the right here, there's an example on the top of a of a tree. And what this model is the dotted lines define time intervals, and it's modeling, it's estimating the size of the population within each interval, and you get a kind of blocky diagram, like the one at the bottom right, which and it's blocky. That it's called a skyline plot, and that's the blockiness is why it's called a skyline. There are various kind of variations of this, most of which begin with the word sky. So you get, rather than saying that your population size is obeyed some kind of a simple function, you can just estimate the variations over time from the data. The standard coalescence, which I've talked about the last couple of slides, assumes, as I said, the population is freely mixing, that therefore all ancestors are equally likely for any individual in the population, which obviously if there's a lot of geographical or some other structure, it is violated. And that can actually cause sampling bias issues if you have some populations that are oversampled. Because if you're seeing a lot of coalescences very quickly, but they're all in a small population, then you're going to think that the population size is small, when in fact they're a confounding effect of population structure. And the extension called, which is a structured coalescent, which splits the population into subpopulations, which are called demes, and that allows for ancestry within demes and migration between them. And you will estimate the individual size of demes and migration rates. I'm not aware in phylodynamics of kind of models that combine the structured coalescent so much with the skyline models. Normally these are looking at mutation rates and assume a constant population size within each deme. These generally work best for small numbers of demes. The models struggle a bit when you get past a few. But one of the questions about this kind of model is what population you are actually studying the dynamics of. But these models were designed originally thinking about breeding populations of organisms. So you can treat the organisms here as the pathogens, but transmission imposes an enormous population structure on pathogens, and here you're completely ignoring it, just saying that any two pathogens can have an ancestor in the previous generation, regardless of who they're infecting. You could also treat them as infections, say that these are infected individuals, but it only works up to a point. Infections don't really reproduce in the way that population genetics expects, and generally these are not models of disease transmission. The parameters like the effective population size are rather hard to interpret in epidemiological terms to get incidence or prevalence or R naught or other parameters of interest. And often you find that researchers will examine a skyline plot by eye for temporal and spatial trends in the effective population size without interpreting the actual numbers. Nevertheless, these models have quite an attractive simplicity, and they're easy to run, and you see them quite a lot. So the example here, this is a study from Mike Worreby in 2008 on the emergence of HIV from Kinshasa. So this is the time tree. It's a time tree on a radial axis, and the grey area goes on to, I think, about 1950. And you can see that it's after 1950 that a lot of the coalescences start appearing. So that's when the population started growing. And you got on the right the skyline plot, and it doesn't look like a skyline anymore, but it's aggregated over lots and lots of trees. So you've kind of taken lots of skylines and averaged them. And you can see that the growth of the population again begins at around 1950. So coalescent models, both the one I've talked about before and the one I'll talk about later, operate backwards in time. The other family of models are forwards time and behave a lot more like the conventional epidemiological model, like an SIR model that a lot of people will be familiar with. What this does mean is that you have to model the process by which samples with which isolates get into your sample. Coalescent models are really reconstructing the history of a sample, so they don't need to necessarily have to think about the sampling process, where in this case you have to have some idea of some probability that a pathogen got into your sample. The sampling has to be modeled, and that's in a way one of the main things that had to be added when adapting the sort of classic infectious disease transmission models to phylogenetics. So a lot of well-known models have been implemented in a phylodynamics framework. There are birth-death models which kind of simply assume that birth is the arrival of a new infection, and death is death or recovery from it, removal from the infected population. That's been done kind of on its own and also with time varying parameters, or like in quite a similar way to the skyline, or with population structure, such as for migration. There are also versions of the SIS and SIR models. So you can see the diagrams on the right. You have a susceptible class that becomes infected and recovered or goes back to susceptible. These are a couple of examples from HIV in Europe. The left is showing temporal trends in some parameters over time. Confusingly, the B, the second one down, sub-figure B, that's what the infected population size from a standard skyline, which is not that interpretable. And it's been using the birth death models. You have the effective case reproduction number, which you can see decreases as the epidemic progresses. You have the rate at which people become non-infectious, which is delta, and that increases once treatment becomes more widely available. And you have a sampling probability, which is, in fact, the researchers here kind of set to what they knew, because a feature of this model is you kind of have to know one of the three parameters, which are a bit more informative to look at than the infected population size. On the right, you have some examples from a couple of Swiss clusters, and these are trajectories in red of the number of sample individuals, and in black by the number of inferred individuals in the whole population, according to a couple of different models of mixing, which are the red and the dotted and dashed lines. So the third category are the epidemiological models that have been the epidemiological structured coalescent. So this is to take the backwards in time coalescent models and reformulate them more to mimic the epidemic models in the previous section. So you're going backwards in time, you're having the same kind of compartmental structures you saw there. And you've done it by kind of adjusting the structured coalescence so that the compartments become the demes. And the demes don't have to be geographical areas, they could be risk groups or something similar. And again, if there were these backward time models, there's no such a need to model sampling. And there are a couple of packages to do this now, which I've listed there. This is an example of one of Eric Voltz's papers from 2013. So this is about HIV transmission from early infection. It was a study in Michigan in the US. So you've got a compartmental model diagram here where infections happen at a rate, and then they go into an early HIV stage, chronic, and then AIDS, all of which can be diagnosed at any stage. And the orange lines represent natural mortality. And this was used to look at the number of transmissions from early and chronic and AIDS risk groups, and from diagnosed and undiagnosed over time. As you see on the right, you've got the number of transmissions and the percentage of transmission. So you can see that the number of the proportion from diagnosed from undiagnosed kind of peaks and then decreases, and proportion from early HIV does the same. So talking about phylogeography, which is basically it infers the movement of ancestral lineages in the phylogeny through space and time, which is related to phyloanatomy, which is something I think is emerging more and more now, which is looking at movement between compartments of a host organism, parts of the body. The structure coalescent is one approach to phylogeography, but it does, as I said before, it struggles with larger number of demes, and it also assumes that you've got a finite number of discrete demes. If you've got better geographical data, if you've actually GPS tagged your sequences, then you'd ideally not want to aggregate those into single locations. So there's another model it's become known as migration, which treats location actually rather like a nucleotide, and as something that mutates over the tree, and that can handle a very large number of states. So there are some sampling bias issues that need to be thought about. There's also a continuous model, which if you do have GPS tagged your samples, you can have you can see how, rather than saying you move between these discrete locations, you can have the lineages diffusing across the landscape. A key difference, I think, between this kind of phylogeography model and the phylodynamics I've talked before, which is that the phylodynamic processes are assumed to generate the tree. You assume that this is kind of the output of a disease model. Whereas here it's more like a mutation. The mutations you see on a tree are usually slightly incidental to the process which driving the epidemic. I'm sure there were cases where that's not true, but normally a lot of mutations are irrelevant to who gets what. So the linear splits are not part of the model that has movement from location to location. It's just something that's kind of carried along with the tree. So a couple of examples. This is from Nuno Faria's paper on the emergence of HIV from Kinshasa. So you've got the phylogeny on the left, which is top to bottom. And these are coloured by location. So you see in the most ancestral sequences for Kinshasa, and it moves around according to the timescale and the map on the right as the epidemic proceeds, as it leaves, it spreads through the country. One enhancement that's been added to the phylogeography models, and as of last year, these are starting to come into structured coalescent models as well, is that you can look at your phylogeny and where, if you've got, say, locations, what locations have a high rate of movement between them, and see what kind of other predictors, epidemiological predictors that matches with. So, for example, this is H3N2 flu from one of Philip Lemay's papers, and they divided the world into communities of air passengers, and showed, using the phylogenetic data, that the passenger flow between these communities was the strongest strong predictor of movement of lineages. You did a similar thing with big and small geographical locations, and for the big locations they got a fairly similar result. For the small, they got more predictors. And this is an example from the original paper about the continuous model. So this looks a bit like the phylogeny kind of imprinted on the map of North America, and that's basically what it is. It's basically you've got you see the phylogeny grow as you move the timeline forward, and you have, by the end, you have the whole tree, and you have the tips appearing, and then also where the process has inferred the internal nodes to exist as well. So you have a kind of way of seeing if your data is suitable, and if the landscape would be suitable. I mean, the restriction with this model is that it's you need to be sure there's not something like a sea in the way, because it doesn't necessarily, or if it does, that you've adjusted the model to know about places that are impossible. But if you have a place where diffusion is plausible, you could use this. The last thing I talk about is transmission tree reconstructions. So what I've talked about until now, they're usually used to infer general properties of the epidemic, such as transmission rates or reproduction numbers. And often they're in situations where you don't have particularly great sampling. Coalescent models assume that sampling is sparse, and forward time models will have an explicit probability of sampling as a parameter. Neither of these are fundamentally concerned, excuse me, with, given the sample you've got, working out exactly who infected who. Whereas there are the category of models for transmission tree reconstruction or source attribution that are more concerned with exactly how the samples relate to you interact in the transmission chain. Many of these assume complete sampling. You have everyone, but that's becoming less true as more methods are developed. Not all of these are phylogenetic or phylodynamic at all. Some are a bit more like the clustering methods in HIV. They just look at genetic distance. There's a list of methods there. Phyloscanner is sort of a method like this, but I wouldn't say it's phylodynamic because it doesn't use time trees, at least currently, and it doesn't fit a model. It's really just looking at the properties of the phylogeny. So on the right, you have, on the bottom, you have, if you colour a phylogeny according to the colours here represent hosts, then you can see, based on where one colour changed to another, who infected who. This is an example from my paper in 2015, which was avian influenza in the Dutch outbreak in 2003. And each host here, rather than being an organism, it's a farm. So every colour at the phylogeny on the left is a separate farm, and you see how this varies on the phylogenetic tree. And on the right, you have the transmission tree, which shows all the farms are individual squares here, coloured by location in the country, and you can then see how that translates to a transmission tree. So finally, I just want to talk a bit about inference, about how you perform these things, these methods, how you form these analyses. So ideally, what you'd like to be able to do is you like to be able to take all your data and put it into one algorithm and get out what you need. The kind of a one-step. You have your sequences, which give you the phylogeny without dates. You have your sampling dates from molecular clock, and you have what other data you want, which allows you to, if you like, locations or risk groups, because you're only going to need them if you want to see how transmission occurs between those groups. That's the ideal. That's the gold standard. Unfortunately, it's not always possible because of limitations to technology. So sometimes you have to find yourself doing a two-step or even three-step procedure to get results. So to go forward, talking about the one-step procedure, I mean, the package you will always hear about for phylodynamics is BEAST, which stands for Bayesian Evolutionary Analysis, maybe by sampling trees, which is certainly the current gold standard one-step package, or packages, which I'll get to in the next slide. And that, because it's a Bayesian method, so as I said before, you get, rather than one tree, you get a set of, a sample of trees from the posterior distribution at the end. You get lots of trees, kind of as a natural consequence of how it works. So it deals naturally with issues of the phylogenetic uncertainty, and it will give you both, simultaneously, both the trees and the parameter estimates for your model. But it struggles with data sets beyond a few hundred sequences, especially for more complex models. BEAST analyses are usually presented, if you would look at a BEAST tutorial, you will usually say you'll take these sequences and then do a one-step procedure. But it's worth knowing that you don't have to do this with BEAST. BEAST can also use a fixed tree for a 2 or 3 step procedure. One note about BEAST, which is possibly confusing, is that there are 2 of them. There are 2 BEAST, which are independent development projects. They can be used interchangeably for a lot of analyses because a lot of them are implemented in both, but there are some which are implemented in one or the other. So BEAST, or BEAST 1, is older. It's, I would say, arguably more user-friendly, and there's more cutting-edge methods for population genetic coalescent models than for phylogeography in BEAST 1, whereas BEAST 2 has more of a flexible modular structure. It's designed to be a bit more like R, so you can download separate modules and run them. And most of the epidemiological models, either coalescent or forward time, they tend to go into BEAST 2 before BEAST 1. For a 2-step analysis, that tends to happen if you can run a fairly standard BEAST analysis and get a time tree or a set of time trees, but your phylodynamic model you can't get that to work. So the procedure there is to build a dated phylogeny or phylogenies with BEAST. So you take your sequences and sampling dates and get a dated phylogeny, but then you have a separate algorithm to do the phylodynamics. Sometimes that's BEAST. BEAST can sometimes do things in 2 stages where it struggles in 1. Sometimes you'll use it to get a set of trees, fix those trees, and then use that to infer the model you're looking at. It's important to realize that BEAST is not just a program to draw trees. But eventually you get to the limit. You get to the hundreds and hundreds of trees where BEAST might give you an answer eventually, but it will take months or months. And then you need a 3-stage process. So then what you need to do, you need to make a phylogeny of the old type, of the type with branch lengths and mutations, using some package, using probably a maximum likelihood package. Then you use a separate package to take that maximum likelihood tree and fit it to a timeline using a molecular clock. And then you use that data phylogeny to fit a phylodynamic model. Step 2, because the speed at which data is becoming available is rather outpacing the speed at which the algorithms are getting faster. There's quite a lot of work being done into step 2 in the last few years. I count now 5 packages to fit your your tree to a timeline. Because you're usually using a single tree here, there's not that much scope for dealing with phylogenetic uncertainty, rather than just taking several variants and running the analysis many times. So in summary, phylodynamics marries evolutionary biology and mathematical modeling of infectious disease. Phylogenies with branch lengths in Canada time are almost always used, and there are some exceptions, but that's fairly normal. The phylogeny is taken to be the history of the epidemic, and by fitting models to that history, we recover important parameters of the epidemic. Phylogeography and source attribution are related areas, which have quite a lot of overlap, but I think are slightly different because they don't have to be phylodynamic. And Bayesian methods are probably the most common. There's the 1-step BEAST for well-behaved data sets and well-behaved models. There are 2-step procedures if you can get some trees out of BEAST, and there are 3-step procedures if you have a very large data set and need to fit it to a timeline some other way. And that's the end of the presentation. So any questions?